ចាសួស្តីយើងចូលមកដល់ section ទី20ក្នុងtopic មួយទាក់ទងជាមួយនឹងnucleic acid ក្នុងផ្នែកនេះគឺយើងនិយាយតែពី RNA ទេដែលcontent របស់យើងហ្នឹងគឺយើងនឹងធ្វើការណែនាំអ្នកទាំងអស់គ្នាទាក់ទងជាមួយនឹងRNA post transcriptional modification ចាហើយយើងនឹងមានដូចជា translation of protein synthesis ចាហើយនឹងmistake calculation of protein synthesis អញ្ចឹងតកតងជាមួយនឹង RNA synthesis process ហ្នឹងយើងដឹងហើយអ្នកគ្រូបានប្រាប់កាលពី session មុនថាវាបែងចែកចេញជា3 pass ដែល pass ទី1ហ្នឹងគេហៅថា initiation pass ទី2ហ្នឹងគឺ elongation ហើយ pass ទី3ហ្នឹងគឺ termination អញ្ចឹងក្នុងដំណាក់កាល3ខាងលើហ្នឹងអ្នកគ្រូនឹងបង្ហាញក្នុងវីដេអូមួយនេះអំពី RNA synthesis ហើយនឹង its processing ពីដំណើរការរបស់វាហ្នឹងថាយ៉ាងដូចម្ដេចអញ្ចឹងបងអាចមើលវីដេអូមួយនេះបាន
So the process by which they can produce enzyme is called the central dogma, which involves from production of RNA from DNA and production of protein from the transcribed RNA. So the important first step is converting the messages inside the DNA into a genetic blueprint, which is RNA. And this step is known as transcription. Now, transcription in prokaryotes could be seen in 3 different phases. And the first phase is initiation, second phase is elongation, and the third phase is termination. So we would go about one by one and look at the details, what are the events in each of these phases. Let's begin with initiation. So in initiation, the key players are definitely the DNA that need to be transcribed, the RNA polymerase, and the sigma factor. So the RNA polymerase and the sigma factor forms the RNA polymerase holoenzyme, and definitely the nucleotide triphosphate, which would be used to synthesize nascent mRNA. So just before the transcription start site, the nomenclature is used as minus to denote the upstream region and the plus to denote the downstream region. At 10 base pair upstream and 35 base pair upstream, there are some well-conserved consensus sequence to which the prokaryotic polymerase can bind. And this binding and finding this promoter is aided by the sigma factor. So the sigma factor really helps in binding of these RNA polymerase to the desired promoter region. Now these sequences in this -10 base pair and -35 base pair are well conserved, and these are known as consensus sequences. Now after that, it has been seen that without the sigma factor, the polymerase can hardly detect the promoter, and even if it detects the promoter and start to transcribe the gene, it would be aborted after transcribing few nucleotides of the mRNA. So without the sigma factor, the fidelity of the transcription is hampered, and at the same time promoter recognition is harder. The most common sigma factor in the bacteria is known as sigma 70. So with the sigma factor attached, the RNA polymerase can properly understand which site to be recognized as a promoter, and it can form the RNA. Now the most common sigma factor is sigma 70, which bind to -35 base pair TTGACA region and -10 TATAT region. Other sigma factors in E. coli involve sigma 32 or sigma 28 for heat shock genes, etc. Now the second step is elongation. Now for elongation step, for the simplicity purpose, this RNA polymerase would be denoted as a bubble. Now RNA polymerase is a big enzyme having beta dash, P beta, alpha, alpha 2, omega, and sigma subunit. Sigma subunit is part of the holoenzyme complex. The active zone is comprising beta and beta dash subunits. So in case of elongation, this RNA polymerase introduces positive supercoiling at the front of it and leaves the negative supercoiled DNA at the back. And inside these enzyme, NTPs enter that would help to form the newly synthesized RNA in a template dependent manner. The strand which is used as a template is known as template strand. The opposite strand is known as coding strand. Now the newly synthesized RNA has a sequence totally similar to the coding strand. Just a difference is the RNA has uracil, whereas the coding strand has thymine. So the sequence in the coding strand is just similar to the mRNA sequence, and the RNA sequence is complementary to the template strand. Now the RNA polymerase move in a 5' to 3' direction and synthesizing the mRNA. And RNA polymerase, the fidelity and the processivity in the RNA polymerase is lesser than a DNA polymerase, because you can clearly imagine if the DNA polymerase makes an error, then that error could be detrimental because replication only happens once. But for a transcription, a gene could be transcribed by a RNA polymerase several times. So one transcript, if it is faulty, it might give rise to a faulty protein, but that might not be so important in the long run. So that's why the fidelity of the transcription process or the accuracy of the transcription process is way lower than the replication, and the rate of transcription is roughly 40 nucleotide per second. Now if we zoom into the structural details of the RNA polymerase in bacteria, and all about these structural details are well known due to the X-ray crystallographic structure, we would clearly see in the beta and beta dash subunit there is the entry site of the untranscribed DNA that enters, and the helicase activity creates the strand, separates the strand, and in a magnesium dependent manner the polymerization happens. Now for polymerization to occur, the specific catalysis is known as metal ion catalysis. Here the magnesium is actually used to stabilize the triphosphate that is getting entered as NTPs from the NTP entry site. Now the template, the newly synthesized strand, which is complementary to the template strand, attacks the phosphate linkage using a nucleophilic attack, and as a result the new template strand is the complementary strand to the template strand. That means the newly synthesized mRNA is elongated. Now definitely there are some antibiotics such as rifampicin which blocks this process by binding to a site adjacent to the active site and works like a steric factor of entry of the substrate DNA inside it. That is how the activity of these enzymes could be blocked by several antibiotics as well. Now then we come to the overview of termination. In bacteria, the termination of the transcription depends upon a termination sequence which is present at the end of these the transcript. Now there are two modes of termination. One is row dependent termination. So row is a helicase which can literally move across the RNA and free the RNA from the hold of the RNA polymerase, and the whole complex dissociates and the synthesized RNA is now free from the DNA. And the second type of termination is known as row independent termination, where it has a palindromic sequence which forms a hairpin loop and it prevents the RNA polymerase to move further forward. And also there is a poly-U site, poly-U stretch, and in poly-U stretch the adenine and uracil bonds are relatively weak, so such that it could be easily broken and the mRNA could fall out of the DNA.
ចាអញ្ចឹងបន្ទាប់ពីបងអូនមើលវីដេអូនឹងហើយបងអូនអាចយកចំណុចនេះទៅពិភាក្សានៅក្នុងថ្នាក់បើសិនជាអត់យល់ថងណាឬក៏មានសំណួរថងណាតែបងអូនត្រូវតែមើលហើយធ្វើការ note ទុកចាអញ្ចឹងដល់ចំណុចនេះគឺយើងនិយាយពី RNA function ចាដែលយើងដឹងហើយថា RNA function ហ្នឹងគឺវាមាន3ទី1ហ្នឹងគឺ tRNA ទី2ហ្នឹងគឺ mRNA ទី3ហ្នឹងគឺ rRNA ចាអញ្ចឹងតកតងជាមួយនឹងតួនាទីរបស់ RNA នៅក្នុងផ្នែកទាំង3ហ្នឹងអ្នកគ្រូនឹងបង្ហាញនៅក្នុងវីដេអូមួយនេះ
There are three major types of RNA which occur in all organisms. They are messenger RNA, that is mRNA, transfer RNA, that is tRNA, and ribosomal RNA, that is rRNA. Let us know about the messenger RNA. As the name suggests, mRNA carries the genetic information from DNA to the ribosomes. Genetic informations on the DNA are transcribed into the mRNA by a process called transcription. Here the message is translated into action, that is, based on the genetic information, different types of proteins are synthesized. The type of gene that is involved in protein synthesis depends upon their strength, kinds, and sequence of nucleotides. It is about 3 to 5% of the RNA content of the cell. The mRNA is always single-stranded. The mRNA is produced as a complementary copy of the DNA which is involved in protein synthesis. Let us know about transfer RNA. Transfer RNA is also known as soluble RNA, sRNA. The tRNA is a small molecule compared with other types of RNAs. It amounts to about 15% of total RNA of the cell. The tRNA molecule performs a number of functions. The most important one is to act as a carrier of amino acid to the site of protein synthesis. There are about more than 20 types of tRNAs. Each tRNA is a specific for a particular amino acid. In bacterial cell, there are more than 70 tRNAs, and in eukaryotic cells, the number is even greater. There are 4 or 5 tRNAs specific for a particular amino acid, and these are called isoacceptor tRNAs. Describe ribosomal RNA. This is found in the ribosomes. The rRNA represents about 40 to 60% of the total weight of the ribosomes. Relatively, it constitutes about 80% of the total RNA of the cells. They are produced in the nucleus. They are the most stable forms of RNA. They consist of single strand of nucleotides. At some regions, the strand is folded.
ចាអញ្ចឹងដល់ចំណុចនេះដែរគឺយើងនិយាយពី mistake of RNA replication ដែលយើងដឹងហើយថាសាំអង់ទីប៊ីយោទិកឬក៏អង់ទីប៊ីយោទិកមួយចំនួនហ្នឹងគឺវាប្រឆាំងទៅនឹងការលូតលាស់របស់បាក់តេរីដោយវារារាំងដោយ RNA synthesis អញ្ចឹងអ្នកទាំងអស់គ្នាអាចមើលវីដេអូមួយនេះបានព្រោះវីដេអូមួយនេះគឺវានឹងបង្ហាញពី mistaking របស់ RNA replication ដោយខ្ញុំលើកឧទាហរណ៍ទៅលើប្រភេទRafapin ណាជាប្រភេទអង់ទីប៊ីយោទិកមួយអញ្ចឹងបងប្អូនអាចមើលហើយបងប្អូននឹងអាចយល់ថាពេលដែលមានការកំហុសនៃ RNA replication ហ្នឹងគឺមានរឿងអីកើតឡើងចាអញ្ចឹងយើងអាចមើលវីដេអូហ្នឹងបាន
So in this video we would look at how antibiotics can interfere with transcription, how that process could be used in molecular biology or in medicine. So let's just first revise the concept of transcription a little bit, then we move to the antibiotics part. So we know in case of eukaryotes, there is a basal transcription factors like TF2 families. All these TF2 families bind the promoter and help to recruit the eukaryotic RNA polymerase. One component of these initiation complex for transcription is transcription factor 2H or TF2H, which actually phosphorylate the C-terminal domain of the RNA pol 2. Now, this TF2H also has a helicase activity. It has a phosphorylase and a helicase activity. Once the C-terminal domain is phosphorylated, it gives the RNA pol as a start signal to move along the DNA and transcribing the mRNA, right? Now let's look at how several antibiotics interfere with these process of making RNA from DNA. First, we'll talk about actinomycin D. Here is the structure of actinomycin D. It's like a pretty flat ring which can get incorporated in between the DNA base stacks, and that can work like a transcription block. So here you can see how the actinomycin D ring gets fit between GC base pairs. So it's worked like a road bump, a huge one. So when the RNA polymerase 2 is moving all across the DNA and trying to transcribe it, it faces this roadblock and it has to stop there. So the transcription is inhibited in a premature state. That is how actinomycin D works. Now, actinomycin D is very important in context of biological research. Imagine, let's say you have actinomycin and let's see what you can do. So let's say you found that these cells, if you give them a magic chemical which you have isolated from some source, let's say, they would really show change in their cell division kinetics and they would show increased proliferation. Now you want to understand that what are the molecular process that leads to this physical change, the proliferation phenotype. So you would think about, okay, transcription could be a reason, their translation could be a reason, post-translational modification could play a role in this. But let's say you have a specific question that transcription is important for that. In order to understand that, what you can do is add actinomycin D to block the transcription, and then see when you block the transcription, you have an effect or not. So if transcription has a role, if it is yes, let's talk about rifampicin. Rifampicin is very specific to the prokaryotic RNA polymerase. It is not specific for eukaryotic RNA polymerase. Eukaryotic RNA polymerase is not sensitive to rifampicin. Rifampicin can bind to the beta dash subunit of the prokaryotic RNA polymerase, and it holds the RNA polymerase there and reduce its promoter clearance probability. Once the promoter is not cleared, abortive transcription can happen and short length mRNAs would be generated, which would not give rise to a functional protein, and that's how the bacteria would be dead. So that is how rifampicin interferes with the transcription process by directly binding with the beta subunit of prokaryotic RNA polymerase. Now, rifampicin do not affect the eukaryotic RNA polymerase. It turns out the eukaryotic RNA polymerase has different, very low binding affinity with rifampicin, and that's why it is resistant. Now, rifampicin, as it can really interfere selectively the RNA polymerase of bacteria, it is used as an antibacterial agent, especially for tuberculosis. Rifampicin is used as a drug in combination with many other
អូខេអញ្ចឹងតកតងជាមួយនឹង session នេះអ្នកគ្រូបង្ហាញតែប៉ុណ្្នឹងហើយតកតងជាមួយនឹងវីដេអូនីមួយៗគឺបងប្អូនត្រូវតែមើលហើយ note ទុកហើយបើសិនជាមានចង្អល់អីយើងអាចយកទៅពិភាក្សានៅក្នុងថ្នាក់បានចាអរគុណ